Okay, on the last slide set, we were talking about the production of the 4 haploid spermatids. So remember, we started the whole process of meiosis after we'd made a copy and the the cell had moved in and become a primary spermatocyte. We've doubled, the everything, the DNA, the cytoplasm, the organelles, and then once we undergo that first meiotic division, we end up with 2 of the secondary spermatocytes, which we eventually divide in telophase number 2 to produce 4 haploid spermatids. We said, but we still have to undergo this maturation process called spermiogenesis in order to produce functional sperm, which have a head, midpiece, and tail. Okay, so we have to rearrange the cytoplasm and we have to form a flagella. So let's get going on this one. So it says, well, what are the 3 regions to a mature sperm? We said again, they have a head, they have a midpiece, and they have a tail. Okay, so let's look at the head. The head is made up of 2 parts. And so the head has this blue, hazy area that looks kind of like a cap. We want to refer to this as the acrosome. I'm going to stop spelling with this mouse. It's going to be the last time. So it has an acrosome. And then it also has a nucleus. The nucleus is underneath the head. So let's talk about the acrosome. I don't know why I jumped ahead so far, but the acrosome houses a ton of these Golgi bodies. And if you remember, Golgi bodies that hang around, Golgi are filled with these hydrolytic enzymes that we call, in this particular case, they're called, the enzymes inside are hyaluronidase. These enzymes are used for egg penetration, so they have to penetrate the zona pellucida that's surrounding that secondary oocyte. And once one of these nuclei get in from the sperm, then that's going to seal that so no other nuclei can enter. Okay, we said the other thing is inside of the head is the nucleus, and that has that haploid set of chromosomes that need to be donated over to the secondary oocyte. Okay, the second thing, we said the midpiece. So here's the midpiece. If you notice, the midpiece has a spiral arrangement. Okay, well, those are the mitochondria. The mitochondria are going to be harvesting glucose or some sugar in order to break it down into carbon dioxide and lots of ATP. So and what is this going to do? It's going to help power the swimming of the sperm cell. And the last thing that these guys need is this flagella. Remember that 9+2 arrangement that we talked about in chapter 4, arrangement of microtubules. Okay, has the 2 doublets running down the center, and that's what gives a whip-like motion to the tail. So we got 9 of these cylinders, 9 that creates this bundle, 9 pairs of doublets that surround 2 in the center. That's a bad looking thing, but anyway, it says when the doublets pass each other, that's what gives a whip-like motion to this tail. Alright. Okay, this is just giving you some of the male genitalia and anatomy. Here's the scrotum, that thin wall pouch of skin. Here's the testes, of course. Well, I put my little blue dot. And inside of the testes, remember, that's where all the seminiferous tubules are. It says, but when these sperm go to mature, finish that spermiogenesis, they go and they sit in this sugary area in the epididymis. And here they complete the maturation process. And And then when a male has an orgasm, these guys are propelled into the vas deferens, which crosses over the pubic bone, goes around the backside of this prostate, and then meets up with the seminal vesicle, which is going to lend to the solutions of semen. So we know that prostate is going to give us 30%, I think. The seminal vesicle is going to give us about 60%. And here's prostate here. Seminal vesicle, I think, is going to give us 60%. And bulbourethra is going to give us around 10% of the solutions that you find. The seminal vesicle and the vas deferens, they join to create what's called the ejaculatory duct. You'll see that out here. Because it's a concomitant organ, remember the guys also urinate out of the same pathway as they ejaculate, so we'll need to shut this off, okay? And so the sperm and semen will be propelled through this tube, definitely not in this position, but anywho. Alright, we know that the bulbourethral gland is going to be responsible for providing the solutions that are going to be necessary to help reduce the friction as that process takes place. Alrighty, so let's move on. Hormonal regulation in male. Well, we know the hypothalamus has ultimate control over the testes. So the hypothalamus releases what we call the gonadotropic releasing hormones. And the gonadotropic releasing hormones, they stimulate the pituitary to actually release the gonadotropic hormones. So the gonadotropic hormones should be your FSH and LH. Okay, so the gonadotropic hormones, again, are the follicle stimulating hormone, or FSH, and then the luteinizing hormone, which is LH. So the follicle stimulating hormone is responsible for helping this process get started, helps to promote spermatogenesis. The luteinizing hormone is going to be responsible for helping to control the production of testosterone by those interstitial cells, okay? So again, we said hypothalamus has ultimate control over the testes, but then it also controls the pituitary. The pituitary gland will be responsible for releasing the gonadotropic hormones. Remember, the hypothalamus is going to release the gonadotropic releasing hormones, which is going to stimulate the pituitary to release the gonadotropic hormones, and the gonadotropic hormones are going to stimulate the organ itself to release certain hormones, or at least some of the luteinizing hormone will. Okay? Now, testosterone is responsible for bringing about as well as maintaining the characteristics that we see in men. Usually men are taller, usually their voices are deeper, usually they have more hair on their bodies, their muscles are bigger. Okay, so they're okay. Oh, generally men also have a pronounced Adam's apple, but generally they have a pronounced Adam's apple. Alright, oogenesis. Now, oogenesis is a little bit different from spermatogenesis in the fact that it must be coordinated with the menstrual cycle. With men, you know, whether that sperm was meant for fertilizing the egg or not, because it may be here, there, everywhere. But these secondary oocytes, they have a specific job to do, okay? So oogenesis must be coordinated with the menstrual cycle, because it's the hormones that we see being produced during the cycle, which are going to be in the ovarian cycle, which are going to be very closely related to every single thing that we see happening in the ovary and every single thing that we see happening in the uterus. Alright, so Again, we said each egg, ovum, secondary oocyte that is being prepared is being prepared for pregnancy. So just like the men we looked at a minute ago, we said that the process of spermatogenesis, it had a specific place. Well, same thing with oogenesis. It occurs within the follicles that are inside of the ovaries. It occurs within follicles that are inside the ovaries. So we know that secondary oocyte or that primary oocyte at first, or primary oocyte that's in that primordial follicle, is going to be the most basic one, and we're going to find that where? In the ovary. Okay, so follicles, they contain the oocytes. At birth, the female has about 2 million. By the time that she's reached puberty, that number has probably been drastically reduced to around 300,000 or so. But in her lifetime, we know, because how many is she producing a month? Bringing to maturity. Probably bringing a few primordial follicles to maturity, but actually only one gets chosen generally to go the mile. Okay, so only about 400 ever mature within a lifetime, because that's one a month. Okay, the woman is not trying to have a litter of kids. One a month. Okay, so females, they have now, the other big thing, females have oogonia until their 3rd or 4th month, embryonic month. So before they're born, all of the oogonia have become oocytes. Unlike men, men have spermatogonia that can undergo mitosis as well as meiosis, so they can produce an almost unlimited amount. Whereas a female, her oogonia have already, prior to her being born, have become oocytes. Okay? So, here is an ovary, and that first yellow arrow is pointing at the primordial follicles, and it says the primordial follicles contain inside a single large primary oocyte, and as they begin this maturation process, they begin producing estrogen. Okay, it says what we want to do is we want to bring a few of these primordial follicles to maturity with each menstrual cycle. Now, even though we're bringing a few along the way with each menstrual cycle, again, only one usually is chosen to go the entire way. So even though we begin this maturation of a few, only one generally is chosen. It says as it undergoes maturation, we move from what we want to say are our primordial follicles to primary, to secondary, to what we call the tertiary or most mature or Graafian or vesicular follicle. This is the one that will actually ovulate. This is all this solution here is referred to as antrum, and it will help to cause that that's supposed to be an M, but I'm having difficulty here. But anyway, it will help to cause the secondary oocyte to be expelled at ovulation. Okay, so the secondary oocyte. One of the things that you can see is when we are producing our secondary oocyte following meiosis 1, there's always an unequal cytokinesis taking place. Okay, so and instead of the males who produce 4 haploid spermatid, the female is going to produce 1 haploid secondary oocyte or egg, and then 3 polar bodies, because it's done by unequal cytokinesis. Okay, so the secondary oocyte that is released at ovulation, we don't consider an egg until it has been fertilized, and this is showing you the fertilization process, which will cause that last polar body to be eliminated, that last amount of chromosomes to be eliminated so that we're in a haploid state. Okay, again, remember we said this is done by unequal cytokinesis in telophase 1, and then unequal cytokinesis again in telophase 2 to produce this one viable egg or ovum in those 3 polar bodies. So what is a polar body? A polar body is this non-functional packet of chromosomes. So in other words, it's a way of eliminating those extra chromosomes, okay? And it does not become an organism, okay? So it's a way of eliminating chromosomes, a way of discarding extra chromosomes.